Today, we are unlocking glycolysis as one of the process that is involved in respiration. Glycolysis. Glyco is to do with carbohydrates. Glyco has to do with carbohydrates from glycogen. So, to do with carbohydrates. Then lysis means to break. Lysis means to break. Now, glycolysis, it is a biochemical process that involves breakdown of carbohydrates. Breakdown of carbohydrates, e.g. glucose into pyruvate. So a carbohydrate like a glucose is broken down into pyruvate. That's very important to understand. Now, it's also important to understand that glucose is a 6-carbon sugar, whose molecular formula is C6H12O6. So it is a 6-carbon sugar, glucose. Now, pyruvate, the end product, it is a 3-carbon sugar, a 3-carbon carboxylic acid. A 3-carbon carboxylic acid. You see how these things they change from a carbohydrate, glucose, which is a monosaccharide of polysaccharides like a glycogen, like a starch and what have you, now to a carboxylic acid, meaning this is not a carbohydrate. From one group of compounds, carbohydrates, to another group of compounds, carboxylic acids. However, what do we want here? We want carbon and hydrogen, basically. That's what we need. Now, this 6-carbon atom, to reduce them to 3 carbon atoms, it means there is a breakdown. But now, throughout this discussion, breakdown is synonymous to oxidation. Okay? Breakdown is synonymous to oxidation. So pyruvate is a 3-carbon carboxylic acid, as we shall see, because its molecular formula, its structural formula is CH3COCOOH. Now that is pyruvic acid, but under physiological conditions, they exist in unoxidized form where hydrogen is not there, but we just have a minus here. Then we call it pyruvate. It is having both the keto group and the carboxylic group. So this one is a carboxylic acid gotten from glucose by its breakdown. Now, whenever I say breakdown, whenever I say the word breakdown, it is synonymous to oxidation. Because if you break something, for example, from glucose, which is a 6-carbon compound, if you break one carbon to remain with a 5-carbon compound, the other carbon has gone with some electrons. Remember, electron loss is oxidation. Good. Now, glycolysis has got 10 reactions. We have got the first five, which we call investment phase. Hope the spelling investment is okay. If it's not okay, you tell me. Then the second one is the second the second 5 or the last 5, if you like. This is the harvest phase, but you all can also feel free to call it payoff phase. Payoff phase, that you invested, now you are harvesting, meaning you are benefiting. Now what happens is that during respiration, as glycolysis is unfolding, in presence of the hormone insulin hormone, which is secreted from the beta cells of the pancreatic islets of Langerhans, what happens? Insulin arrives to respiring cells and causes insertion of GLUT4 transporters. GLUT4 transporters. GLUT is for glucose transporters, but glucose transporter type 4. There are different types of GLUT transporters, of course, but type 4 is very common, especially muscle cells and liver cells, or hepatocytes, if you like. Now, glucose comes and enters into the cell through GLUT4 transporters. It comes and enters there. Now, as it is entering in the cell, this glucose you see here, as it is entering the cell, what happens is that it arrives in the cytosol. Feel free to call it cytoplasm, but also you can feel free to call it cytoplasmic matrix. Now, in the cytosol here, there is an enzyme which have to commit glucose to glycolysis. How? By activating glucose. Now, in science, in biochemistry, whenever you add a phosphate group on any substance, whether a protein or whatever, you are activating that substance, because a phosphate group has got a negative inductive effect. It has got a tendency of attracting electrons from a system onto which it is bonded, making that system a little bit electron deficient, meaning it can easily be attacked by any available nucleophiles. Therefore, it becomes very stable. So now it becomes very unstable. Now what happens is glucose is phosphorylated, but the phosphorylation is in position 6, glucose 6-phosphate. Now, that phosphorylation has got 2 functions to play. The first one, to commit glucose to glycolysis. That is one. The second one is to do what? To make it chemically very reactive. The third one, to trap glucose not to exit the cell, because a non-phosphorylated glucose molecule can easily cross and go back to the tissue fluid or to the blood, if you like, and what have you. So what happens is that glucose is now committed here. It has been activated by phosphate group added. Secondly, it has been done what? It has been committed to glycolysis. Then it has been entrapped or imprisoned inside the cell. Now what happens is that the source of the phosphate group is ATP, meaning ATP which is available, it loses a phosphate to form ADP. In other words, adenosine triphosphate loses a phosphate to form adenosine diphosphate. Now, the other one phosphate from ATP, it goes and joins glucose at position 6 to form glucose 6-phosphate. That's very important to understand. But this process is enzyme catalyzed, mind you. Make no mistake to think that it is just spawn, whatever. It is enzyme catalyzed. Who catalyzes that? It is an enzyme hexokinase. An enzyme hexokinase catalyzes that process. But hexokinase is in all cells that respire using glucose, whether brain, whether muscle, whether liver, whatever, everywhere, whether skin cells and what have you. However, when it comes to some tissues of organs, for example, those of the liver cells, hepatocytes, have got another enzyme called glucokinase. Glucokinase. That enzyme, glucokinase, it is not that name is not another name for hexokinase. It's another enzyme, but which is an isoenzyme to hexokinase. So that isoenzyme does what? That isoenzyme is more potent than hexokinase. In other words, it has got a higher turnover number than hexokinase. Master, what is a turnover number? Turnover number refers to the amount of substrate that can be transformed into products by an enzyme at a particular time. So this one can convert very many glucose molecules into glucose 6-phosphate per unit time than hexokinase. So the liver, why does it have that? Remember, the liver is the second most diversified organ next to the brain in the body. Therefore, and secondly, it is the largest visceral organ with a lot of about 13 parallel functions. So it needs a lot of energy. It is involved in a lot of biosynthetic reactions. So it needs a lot of energy here. Now what happens is glucose 6-phosphate now has no choice of returning back. It is now going to continue. However, glucose is an aldo sugar. Why don't we? Now, glucose being an aldo sugar, it is converted to another isomer, to an isomer of it, which is even more reactive, which is a keto, which sugar, each sugar is fructose. So what happens is glucose here is converted to fructose. Which fructose? Fructose 6-phosphate. Fructose 6-phosphate. Now it is converted there. It is converted into fructose 6-phosphate. However, that kind of reaction is enzyme catalyzed. Who catalyzes that enzyme? It is the enzyme. It is the enzyme hexose isomerase. It's an enzyme hexose isomerase. Remember, isomers have got the same molecular formula but with different structural formula. Now what happens is that hexose isomerase catalyzes conversion of glucose 6-phosphate to fructose 6-phosphate, meaning fructose 6-phosphate is chemically more unstable than glucose 6-phosphate. Master, you sound as if glycolysis loves more unstable intermediates than stable ones. Master answers yes. Why? We want to break them. So we cannot break something which is stable. We can break something which is very stable. That is the answer from the teacher. So what happens is fructose 6-phosphate is further phosphorylated to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Master, why not diphosphate? Diphosphate means two substituents, two phosphates in the molecule. Bisphosphate also means two, but bisphosphate means one is from the one end of the molecule, the other one is on another end of the molecule. The distant spacing, that's whatever. So that is a language of biochemistry need to be used too. Fructose 1,6-bisphosphate. This is now even more unstable. Why? First of all, it is a keto. Secondly, it has got two phosphates. Two phosphates now can disturb this molecule more than one phosphate. Now, which enzyme catalyzes that? This enzyme catalyzing that whatever is what? This enzyme catalyzing that whatever is phosphofructokinase 1. Phosphofructokinase 1. It is that enzyme. You can also simply call it PFK1. Now this enzyme, there is also phosphofructokinase 2, but it has got another function like this one here. So phosphofructokinase 1 catalyzes conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. But then, where does this second phosphate come from? This second phosphate also comes from hydrolysis of ATP. So ATP is also again invested to ADP, and the inorganic phosphate joins here to get fructose 1,6-bisphosphate. But now, look here. So what happens is that the fructose 1,6-bisphosphate you see here, members, the full video will be on YouTube. You click in whatever there on my profile there, TikTok, you'll see my link. You hit it there, a link will take you to YouTube directly, and there will be play there in respiration. So what happens is the fructose 1,6-bisphosphate, because of it having two phosphate molecules, phosphate groups within its structure, it is now even chemically more unstable. So it breaks down into two intermediate C. It breaks down into two intermediate C. That's very important. So what happens is, which intermediate? The first intermediate is what? The first intermediate is glyceraldehyde. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Then to dihydro. Then another one is dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, members, for the first time we have 3 carbon compounds, but they are also sugars, because carbohydrates are either aldehydes or ketones, only that they can either exist as polyhydroxyaldehydes or polyhydroxyketones, but these ones are the simplest sugars now. So glyceraldehyde, it can be gotten from our glycerol, so it is a 3 carbon whatever. So aldehyde, meaning it is having an aldo group. So we have got a hydroxyl group, a hydrogen, then a hydroxyl group, a hydrogen. So what happens is that this side here we shall have what happens is that we shall have a phosphate group here. We shall have, let's have hydrogen here, then here we shall have a phosphate group here, double bond or whatever, or minus. So we have our phosphate group here. So this is our glyceraldehyde 3-phosphate, meaning this is carbon atom 1, carbon atom 2, carbon atom 3. Okay, glyceraldehyde 3-phosphate. Now it is a 3C sugar. Now this one is the hydroxyacetone phosphate, so it is having a keto in the middle there, keto group there. So dihydroxyacetone phosphate. We can choose to put a phosphate this way, or so that one becomes our phosphate group, bonded there, only there. So this one is our hydrogen. So here, hydroxyl group, hydrogen. So you see, this is a 3 carbon sugar. This is a 3 carbon sugar. So look here. Now, this intermediate here. Now, for fructose 1,6-bisphosphate to split, there is an enzyme that catalyzes that. The process is enzyme catalyzed. Which enzyme catalyzes that? Is aldolase. This enzyme favors splitting of this molecule here, fructose 1,6-bisphosphate, to these two intermediates. Master, why is aldolase? The reason is why is it that it is aldolase? Is that it is glyceraldehyde 3-phosphate. It is having an aldo sugar. It is more stable than this one. It is more it is more stabilized than this one here. This intermediate is not favored if respiration is to occur. So what happens is that since this one is not favored for respiration to occur, it means it is going to be converted to glyceraldehyde phosphate. Which enzyme? The enzyme is triose isomerase. Triose isomerase. The enzyme becomes triose isomerase. Hope that one is understood fantastically. Now look, if you can count, this first reaction here is reaction 1. This is one here is reaction 2. This one here is reaction 3. This one here is reaction 4. This one here is reaction 5. Remember we said that the first 5 are investment phase. Why investment phase? We have invested there 2 ATP molecules. Hope that one is understood very, very well. But now, before we proceed, I want you to note this. Reaction 1 and reaction 3 are irreversible reactions. The rest are reversible, meaning, for example, reaction 2 here, the same enzyme catalyzing the forward reaction, it is the same catalyzing backward reaction, meaning the same reaction can catalyze the conversion of this one here back at this one here, the same enzyme. This reaction here, you see here, of the aldolase, it is also reversible. The same enzyme, aldolase, can catalyze splitting, but it can also catalyze the combination of these two back to the other one there. Okay, that's very important. When would that one be more even important to help us while discussing gluconeogenesis somewhere where we are heading? Now, that is it. Those are the first whatever. Another take home we need to know. You see, the first enzyme was hexokinase or glucokinase, another glucokinase, then isomerase, that one, whatever. Now, you see, hexokinase reaction, the enzyme for reaction 1, hexokinase, and the enzyme for reaction 3, phosphofructokinase 1, all of them end with the word kinase. Now, kinases, whenever in your journey of biology, whenever an enzyme ends with kinase, whenever an enzyme ends with kinase, it means that enzyme catalyzes addition of a phosphate group on the compound. That's why this one was, these are hexokinases because they catalyze additional phosphate on glucose. This one is also kinase because it catalyzes additional phosphate on glucose, on fructose 6-phosphate. Now, the reverse reaction, the enzyme that catalyzes the reverse reaction will be a phosphatase. So, in nomenclature of enzymes, whenever you find an enzyme, its name ending with phosphatase, it catalyzes the removal of a phosphate. Okay? That is very important to understand. Now, we proceed. So we are done with the investment phase. But before we go, you need to notice that this now, it's as if we have 2 molecules of glyceraldehyde 3-phosphate, because dihydroxyacetone phosphate has been converted into glyceraldehyde 3-phosphate. Now from now on, we're going to continue with the 2 molecules, meaning our arrow is going to head downwards. But now I'm going to start from here. So now here, what are we going to have? This glyceraldehyde 3-phosphate is going to be converted into now, within the entire journey of respiration, while oxidizing glucose, this is where we end to talk about carbohydrates. Carbohydrates ends here. The rest, we're going to be dealing with carboxylic acids. Now, note that. Now here, we're going to form a very beautiful compound called what? The compound is called, this. Remember, this is a continuation of here. Now, we're going to form a compound called what? Glycerate 1,3-bisphosphate. Glycerate 1,3-bisphosphate. Bis meaning 2 molecules, 2 phosphate groups on both ends. Yes, glycate 1,3 bisphosphate. Now look, we have got glyceraldehyde 3-phosphate, meaning it is having one phosphate group substituted there. Now comes that here we have bisphosphate. Where is the origin of the next phosphate group? Now that phosphate group, I won't delay you, that phosphate group comes from the cytosol, comes from the cytoplasm. It is just an inorganic phosphate, not from hydrolysis of ATP. But remember, this is an aldo compound, an aldehyde group to form a carbo, because here what we have a carboxylic acid. Look, we have got glycerate 1,3 bisphosphate. What does it mean? It means that we have got a carboxylic acid. Now what happens is that here we have got carbon. So meaning glycerate 1,3 bisphosphate. It means here we are going to have a carboxy whatever, but where a phosphate group is located. You see, glycerate 1,3 bisphosphate. Now that is it. Now this is carbon atom 1, 2, 3. Now what happens at carbon 3? We also have a phosphate group there, joined by a phosphoester bond. So what happens here? We are going to have hydrogen. We are going to have an OH group. We are going to have hydrogen. You see? Now the functional group we have here is an aldo group. You see? We have an aldo group. Are you seeing that aldo group there? Are you watching that aldo group there? The functional group we have here is an aldo group. Are you seeing it? For carbohydrates. Now here the functional group we have is a carboxylate group or a carboxylic acid group for carboxylic acid Z. So that one is very important to understand. So what happens is that this process here, to form a carboxylate group, we need to steal hydrogen from here and fix oxygen there. What happens there? First of all, we need to steal this hydrogen there. Who is the fifth stealing the hydrogen? The fifth is called oxidized NAD, oxidized nicotinamide adenine dinucleotide. Now, oxidized nicotinamide adenine dinucleotide, oxidized NAD, converts to reduced NAD. If you can even write it like that, whatever if you like. Sometimes one hydrogen, whatever. Now converts to reduced NAD. Now at reduced NAD, this reduced NAD or reduced nicotinamide adenine dinucleotide, reduced nicotinamide adenine dinucleotide is now carrying hydrogen, but hydrogen is stolen from this compound here. So what happens? It means now we are forming a carboxylate group. But master, does the reaction occur spontaneously? Master answers no. It is purely enzyme catalyzed. Which enzyme now? The enzyme is going to be glyceraldehyde 3-phosphate dehydrogenase. It's called glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase means it catalyzes stealing away of hydrogen from glyceraldehyde 3-phosphate. So from a carboxylate whatever here. So that is the enzyme catalyzing that one there. So what happens is we can now proceed. Now what happens is that this compound here, now we have to be happy. The reduced NAD can now be shunted into the mitochondrion if conditions are favoring to liberate for us ATP. It can only give you energy if it enters the mitochondrion in the electron transport chain, where it will have to be oxidized by the protein called reduced NAD dehydrogenase or complex 1 of the electron transport chain, as we shall see where we are heading. Now what happens is that This glycerate 3 phosphate, glycerate 1,3 bisphosphate is now converted to glycerate 3 phosphate. Where has the phosphate gone? Where has the phosphate gone? Now, the phosphate in position 1. Remember, the inorganic phosphate had been added in position 1 here. This is position 1 here, sorry. It had been added here. So what happened is, it is now going away. What is happening is that ADP comes and combines with that inorganic phosphate to form ATP. Now, this kind of formation of ATP is called substrate level phosphorylation. Nikaichwa emphasizes that substrate level phosphorylation. Substrate level phosphorylation. What does that one mean? That one means formation of ATP. Substrate level phosphorylation here means formation of ATP by combining an inorganic phosphate with the ADP to form ATP. Without going through the electron transport chain. Okay, no need of the electron transport chain here. Meaning, you see, it is even formed without reaching the electron transport chain, not even reaching the mitochondrion. We have not yet entered the mitochondrion. Glycolysis occurs in the cytosol or cytoplasm of a cell. Okay, so now we have formed the first ATP, but I don't want you to forget the stoichiometry. We have 2 molecules of glyceraldehyde 3 phosphate. Now what happens? It means that even the glycerate 1,3 bisphosphate, there are 2, meaning everything is times 2. So also 2 ATP molecules are formed here. Here 2 reduced NAD are formed here. Don't forget that kind of that stoichiometry. Don't forget that stoichiometry, members. So what happens is that here, glycerate 3 phosphate, we have also 2. So what happens is that this glycerate 1,3 bisphosphate is now the enzyme catalyzing this process here is called glycerate isomerase. It's called glycerate is called the glycerate. This enzyme is called glycerate kinase. We shall call it glycerate kinase. So glycerate kinase, meaning it is the same enzyme which can catalyze even additional phosphate on this one here. Okay, it can catalyze it is 2-way glycerate kinase. So what happens is that this glycerate 3 phosphate is now converted to glycerate 2 phosphate. You see, glycerate 2 phosphate. What is happening here is that the phosphate in position 3 here, is taken to position 2 by which enzyme? By glyceromutase, by phosphoglyceromutase. Phosphoglyceromutase. Why mutase? It's like it is catalyzing a mutation, just a sudden change in the structure from position 3 to position 2. Why are we shifting the phosphate from position 3 to position 2? There is something we want to do there. We want to introduce an enolase group there. So what happens? The second step here, we are going to form a compound. Phosphoenol pyruvate. We're going to form a compound phosphoenol pyruvate. we are going to form a compound phosphoenolpyruvate. We are going to form a compound phosphoenolpyruvate. Now that phosphoenolpyruvate you see there is formed as a result of the enzyme enolase. It introduces an enol group. So phosphoenolpyruvate. That's what happens. Phosphoenolpyruvate. So what happens is that this phosphoenol, to give you a picture here. Now, this phosphoenol, what happens is that at phosphoenol level here, the phosphoenolpyruvate, okay, we are going to have a double bond there. So pyruvate. So here, what happened? Phosphoenolpyruvate. Here, we are going to have a double bond there. Now here, we are going to have pyruvate. We are expecting a carboxylate there. We're expecting a carboxylate there. Now, phosphoenolpyruvate, what happens is that at this position here, we are going to have a phosphate group there. Of course linked by a phosphoester bond. So I'm going to have a phosphate group there. Okay. So what happens is that at this point here, we shall have a hydrogen, but carbon still yearns for more. Now we're going to have a hydroxyl group there. Okay. Now this group you see here is an enol group, but having a phosphate there. Why we have been catalyzing, we want carbon atom 2 to be catalyzed because we are going to form to introduce a keto group there to form pyruvate. So what happens is that that reaction is catalyzed by an enolase enzyme that introduces an enol group. But look, where has the hydroxyl group come from? In chemistry, we don't destroy matter. So that means, what does it mean? It means that there is addition of a water molecule here. We add a water molecule there. Okay? The reaction is a hydrolytic reaction. It involves addition of a high water molecule there in the process. That's the origin of this hydroxyl group there. Don't forget. Learners normally forget this water molecule there. Never forget that. If you're following Biology King Yugi, then you are nurtured to become one of the best scientists, both on planet Earth and on planet Mars. So what happens is that here, this phosphoenolpyruvate is now going to be converted into pyruvate, a 3-carbon compound, pyruvate, a 3-carbon compound. So, which enzyme catalyzes that? Now, to convert this one to pyruvate, you have to form ATP. So, what is happening? Here? ADP comes and steals a phosphate group to form ATP. Remember, everything is times 2. So this one is times 2, okay? But meaning also this one will be times 2, what you see here. So then also pyruvate will be times 2. So our pyruvate is formed now here. We have our CH3, C double bond O, then C double bond O, C, or whatever. So that is our pyruvate. Under physiological conditions, they are done what? They are ionized. Now, something to note here. Now, look, when you look very well here, what happens is that we ended. This one was reaction 5. The investment phase where I invested 2 ATP molecules because everything that was times 2, as we saw. This is the times 2 ATP molecules. Sorry, the times 2 begins at reaction 6. Now, what happens is that here we invested at reaction 1 an ATP molecule, then we invested at reaction 3 and another ATP molecule. Those are the 2. So what happened? What are we saying here? Now, the thing is that here, the harvest phase or payoff phase, it begins with the reaction 6. The reaction 6 is this one here. This one is the reaction 6 here. Then from there, we went to reaction 7 here. From there, we went to reaction 8 here. From there, we've gone to reaction 9 here. Then finally to reaction 10. This reaction 10 is remember we said there are 10 reactions. If you recall where we began, reaction 10 is catalyzed by the enzyme pyruvate kinase. Pyruvate kinase, also famously called PK, pyruvate kinase. When you say PK, you are meaning this enzyme here. Of course, if you are in the respiration as a topic. So that is the point. So these are the 10 intermediates of glycolysis, of the glycolytic pathway. Now, having formed 2 molecules of whatever, there's something I want you to note here. What is the investment? What have we invested? We invested 2 ATP molecules. Then what have we harvested? We have harvested 2 reduced NAD molecules. These ones are very good if they enter the electron transport chain. They got form for us ATP. Then, but sometimes they may not enter, depending on the conditions, as we shall be finding out some other discussions later on. So we have also harvested 4 ATP molecules. 4 ATP molecule. The first 2 were at reaction 7. Then the second 2 at reaction 10. Okay? Now, those are the 4 ATP. Remember, we had invested 2. Now you know the net ATP of what? Now, total profit, 2 reduced NAD and 2 ATP molecules formed by what process? Substrate level phosphorylation. Whenever you form ATP before reaching the electron transport chain, that ATP has been formed directly, is formed by substrate level phosphorylation. The other one in the electron transport chain, we shall call it oxidative phosphorylation, but here substrate level phosphorylation. The other one is formed as a result of redox reactions, as we shall be seeing later on. So members, this is our glycolysis. Feel free in the comment section to show us your views. Where do you think, what can you contribute on this entire process? What is not clear to you? We shall come back and correct and whatever. Feel free to contribute. Feel free to criticize. Of course, on TikTok, I post a short video, but the full video is on YouTube. With that, I remain Biology King Yugi. I thank you very, very much. Thank you for your attention and for your time.